alcohol oxidase biosensors. Alcohol oxidase biosensors are those biosensors in which alcohol oxidase is used as a biorecognition element. Alcohol or ethanol is one of the products which is produced in our different metabolic cycles. Alcohol oxidase is responsible for the conversion of low molecular weight in alcohols into their aldehydes and respective ketones. It is a flavin adenine dinucleotide dependent enzyme. It utilizes molecular oxygen. The assay or the estimation of the alcohol oxidase is based either on the consumption of oxygen or the production of hydrogen peroxide. Alcohol oxidase enzymes could be divided into 4 types depending upon the substrate which upon it is acting. इन 4 types में पहली type short chain alcohol oxidase, second type is long chain alcohol oxidase, third is aromatic alcohol oxidase, and the fourth is secondary alcohol oxidase. अल्कोहल ऑक्सीडेज एंजाइम अपनी स्पेसिफिसिटी के लिहाज से अपने सबस्ट्रेट पर एक्ट करते हैं. नाउ वी लुक एट द जनरल स्ट्रक्चर ऑफ अल्कोहल ऑक्सीडेजेस. Alcohol oxidase is a key enzyme for the metabolism of methanol. It has a molecular weight of 600 kilodalton and is a large size protein. Alcohol oxidase consists of 8 identical subunits, and all of these identical subunits have an ability to carry one non-covalently bound flavin adenine dinucleotide molecule, and FAD basically act as a prosthetic group. This is required for the biological activity of the molecule. आप इस स्लाइड में अल्कोहल ऑक्सीडेज का स्ट्रक्चर देख सकते हैं. This is a 3-dimensional structure and it is composed of alpha helices and beta pleated sheets. You can see the FAD molecules in ball and stick model in the slide. Ethanol biosensors. Ethanol biosensors basically used alcohol oxidase. We know that ethanol is an important metabolic product and it is also an important industrial product. Different industries relies on the production of ethanol, such as health and diagnostic, food industry, medical science, agriculture, and environment. In medical science and diagnostic, the amount of alcohol is determined at cellular level to determine the different pathological or physiological conditions, about which we will discuss at the end of this module. Ethanol biosensors are based on alcohol oxidase, and these biosensors are reported to be stable and practical, and they are successfully being used in the industry. Now, I will give you 2 examples of the alcohol biosensors. The first is the automatic biosensors for ethanol detection. Alcohol is produced during the industry during different processes, and the sensor is based on alcohol oxidase, and in this type of automatic system we use 2 types of enzyme. One is alcohol oxidase and other is reddish peroxidase. You can see it in here. Alcohol oxidase basically oxidized alcohol into acetaldehyde, and the hydrogen peroxide which is produced as the result of this catalysis is utilized by reddish peroxidase, and that is detected by the electrode as a signal. This small automatic bioanalyzer is successfully used in industry for the determination of alcohol in process technology. Now, a second example, and that is very interesting example, in which we use alcohol oxidase biosensor to determine to check the brain activity of a rat. This biosensor is used in vivo, यानी कि it is used within, हम इस biosensor को brain में इस्तेमाल कर सकते हैं. And this biosensor basically determines the hydrogen peroxide and the interfering agents which are present in the brain are removed by using certain membranes. Now we will look at the strategy that how this biosensor is produced. In this biosensor, the enzyme alcohol oxidase is used, and to stabilize the enzyme, BSA, bovine serum albumin, is used. You can see it in here in green color. This enzyme is capable of hydrolyzing alcohol into its components and produce hydrogen peroxide. This hydrogen peroxide is state determined by the electrodes. इस बायोसेंसर में हमने सिल्वर का रेफरेंस इलेक्ट्रोड इस्तेमाल किया, जबकि प्लेटिनम बेस्ड सेंसिंग इलेक्ट्रोड्स को इस्तेमाल किया गया. This electrode also has an epoxy tip that was about 200 micrometer in size. This biosensor was very successful is determining the brain activity of a rat. So the enzyme based alcohol oxidase based enzyme could be used for determining the biological activity of a cell in vivo. In summary, we can say that alcohol oxidase is a very important enzyme for the fabrication of biosensors. It is a flavin adenine dinucleotide dependent enzyme. The biosensor based on alcohol oxidase are used in industry to check the process technology, and they are also very useful for the determination of biological activity in vivo. For example, a biosensor is fabricated to determine the biological activity of a brain using the alcohol oxidase.